tayo. Okay, so yan, recording na. So for this chapter, we are going to talk about amino acids and peptide bonds. No? So these molecules, or these are the fundamental biomolecules in our body because amino acid is basically the monomer, no? Siya yung pinaka-smallest unit ng lahat ng mga molecules sa ating katawan, no? Lahat ng other biomolecules in our body. Okay? So, let's talk about the chapter outline. So, ano yung mga di-discuss natin? So, we will see ano yung importance ng amino acids. Then, ano-ano yung mga amino acids, no? Actually, 20 sila mahigit, no? So, alamin natin kung ano yung characteristic nila, properties nila, no? And then, ano yung behavior ng amino acids, no? So, amino acid can act as both acids and bases, no? So, tignan natin ano meaning nun, no? And then, later on, how do amino acids combine to form proteins, no? So, paano sila nagsasama-sama to form the peptide bond, okay? So, let's continue. So, ang amino acid, no? Isa lang ito sa maraming molecules sa ating katawan that allows our life na involved sa ating sa ating mga cells, no? For example, yung mga red blood cell, protein lang yan, no? White blood cells, no? And then lahat halos ng type ng cell may protein yan. Okay? Ang isa pang biomolecule sa body natin is the carbohydrate, no? So pag sinabing carbohydrate, ito yung mga sugars, no? Yung mga sugar na yan, yun yung ginagamit ng ating katawan, no? To produce energy. And sometimes yung sugar, they are stored, no, para maprotektahan yung ating cell, no. For example, yung mga fats, no. Yung lipids naman, these are fatty molecules, no. So sila yung mga tumutulong to maintain the integrity of the cell. For example, yung cell membrane. Sometimes yung ating lipids, they serve as the hormones, no, sa ating katawan, no, such as the estrogen, testosterone, no, mga lipids yung mga yon. And lastly, nucleic acid. So itong nucleic acid, this is the smallest portion sa ating genetic sequence, yung ating DNA, no. Okay. So yan. So yung apat na biomolecules na yan, may di-discuss natin yan for the following weeks, no, sa ating semester. So for now, punta muna tayo sa pinaka-basic, no, yung proteins, no. Okay. So yung ating proteins, ito yung itsura niya. So our proteins are composed of the smallest unit called amino acids, no? So yung amino acids, they are joined together by peptide bonds. So yan, magiging malaking protein na yung ating molecule, no? So yung carbohydrates naman, ang smallest unit niyan ay sugars, no? Ang nucleic acids naman, ang smallest unit niyan ay yung mga nucleotide bases natin, no? Yung ATGC or AUGC, no? And yung lipids, ito yung mga fatty molecules, no? Yan. So let's focus on amino acids, no? So when we say amino acids, these are the monomers of our protein. So ibig sabihin, sila yung smallest unit ng proteins natin. And there are generally 20 types of common amino acids, no, found in our body proteins. Although there are other amino acids na uncommon na nag-exist din sa body natin. Also, what's something weird with amino acid is that those amino acids in L configuration are used in living organisms, no. Yung D na configuration, hindi siya yung ginagamit sa organisms natin. Ito yung weird na na-discover nila sa sa Miller-Urey experiment na. Kung naalala niyo pa yung Miller-Urey experiment, what happened there is that, 'di ba, may sa isang container ni-react nila yung iba't ibang molecules, no? Nakakita sila ng amino acids doon. The problem with amino acids that they found there is D yung configuration niya, no? So, dextrolevotary, no? So, ibig sabihin, clockwise yung rotation ng molecule. So, yung kapag nabasa nyo yung D and L, it has something to do with chirality lang, no? Sa orgchem. So, kapag D, right-handed yun, no? Okay, so, ano ba napansin nila dun sa Miller-Urey experiment is puro D amino acids yun na po-produce, no? Yung nasa tao, L eh. Okay, so ibig sabihin, pwede kang makagawa ng amino acids, yet life is weird, no? Hindi ka pa rin talaga technically makagawa ng life sa laboratory in an instant, okay? Because of the conformation of the amino acids na nabuo doon. Okay. And then, humans are incapable of synthesizing half of the 20 common amino acids. No? So, kaya meron tayong tinatawag na essential amino acids. No? So, when you say essential amino acids, these are the amino acids that cannot be synthesized by the body. No? So, in order for us to get those, no, we have to obtain them from our diet. No? So, we have to eat protein-rich food, no? Such as fishes, no? Lean meat, no, and then sometimes gulay, no, so they provide us proteins, no, or they provide us the essential amino acids, no, for us to be able to produce different types of molecules, no, in our body. Okay, so the general structure of amino acid looks like this one, no? So it is a molecule which has 4 different groups attached to it, no? Mostly 4 different groups attached to it, no? Except for glycine. So ito yung itsura ng ating molecule. So this is your central carbon. So this is your alpha carbon. You have your amine group. So when you say amine group, that's NH3 or NH2. Okay. You have your hydrogen there. You have your carboxylic group or carboxyl group, and you have your R group, no? So when we say R group, no, parang orgchem lang to. Pag sinabing R group, iba-iba yung pwedeng kumabit dyan, no? So pwedeng hydrogen, pwedeng oxygen, and so on and so forth, no? Pwede iba't ibang group ng atoms yung kumabit dyan. So, dito nagkakaroon ng changes, no? Dito nagkakaroon ng uniqueness yung ating amino acids, no? Because since iba-iba yung atoms or molecules na pwede kumabit dito sa R group, then we can create lots of types of proteins, lots of types of amino acid talaga, no? Papalitan mo lang yung R group ng iba't ibang molecules, no? Okay, so yan. And the geometry of the amino acid is tetrahedral. Okay, so the carbon in the center is sp3 hybridized. So it assume a tetrahedral structure na. Okay, so ibig sabihin may 3D shape yung ating amino acid. Okay, there are 2 types of amino acid that exist, no? So depending to sa rotation ng ating molecule. So we have the L or the levorotary. Okay, so that is the left-handed molecule. So that means it turns counterclockwise. And then we have the dextrolevotary or the D amino acid. That is the one that rotates clockwise. No? Okay. So yan. So ang pagkakaiba lang yan is that yung orientation nila ay magkaiba, no? So they are chiral to each other, no? So when you say chiral, kung naalala pa sa orgchem, mirror image lang sila. So basically, the two are enantiomers of each other. Okay? So ganun lang. Although di na natin i-focus masyado yan, ha? Basta alam lang natin, for each amino acid, dalawa yung version niya. So one is the L, the other is the D. Yun nakikita natin sa living organism, L configuration yan. Okay. While yung D, sila yung hindi nakikita sa living things, no? So yan. So there are 20 types of amino acids that we have to be familiar, no? So, hindi ko na sasabihin yung remember kasi hindi naman tayo face-to-face classes, no? Pag face-to-face class kasi pinapakabisado ko talaga to. Pero for now, we can, we can use the cue cards na lang or we can use the PowerPoint while reviewing for this topic na. So there are 20 common amino acids that we have to remember, and depending on the R group, no, we can classify them as aliphatic, aromatic, acidic, basic, and di pa pa, no. So yan. So tignan natin yung mga uri ng amino acid. So ito yung mga 20 na to. Ideally, kinakabisado to, pero sa ngayon, wag nyo nakabisaduhin na. Be familiar na lang kung ano itsura nila. Okay? So we have the alanine here. So ito yung general structure ng amino acid, ha. So this is the alpha carbon, the nitrogen group, the carboxylic group, and the hydrogen is located at the back. So ito yung R group. Ito yung pinapalitan natin. So yun, iba-iba yung R groups natin per amino acid. For example, for alanine, we have the CH3 R group, no? So we have the methyl group, no, attached to your alpha carbon here. You have the glycine. So sa glycine naman, instead na isa lang yung R group mo, wala kang R group, no? So parehas hydrogen yung nakakabit sa alpha carbon. And glycine is the only amino acid that doesn't have chirality center. No. Bakit? Kasi pag sinabi natin chirality center, that means there must be 4 different groups attached to your atom or your carbon. No? In our case, for glycine, that's not true, no? Kasi ano yan eh, dalawang hydrogen yung nakakabit sa kanya. Therefore, it is not a chiral center. And yun, so just change your R groups, marami kang mabubuo na molecules, no? Marami kang amino acids na magagawa. Okay. So let's take a look at them per group, no? Per group na natin siya tignan ngayon. So let's start with the nonpolar aliphatic R groups, no? So when we say nonpolar aliphatic R groups, these are R groups na carbon chain or hydrocarbons lang tong mga to. Okay? So yan. So we have 7 yan na. So we have 7 nonpolar aliphatic amino acids, no. One is the glycine, alanine, proline, valine, leucine, isoleucine, and methionine. Okay. So ano mapapansin nyo sa R groups natin, yung highlighted in pink color. So what we can see is that yung ating highlighted na groups ay puro carbon nga, no? Carbon, hydrogen, and sulfur. So those atoms are nonpolar, no? Let's say. Okay. So yan. So these R groups meron silang contribution sa hydrophobic interaction. Okay. And then yan, they help to determine the 3D structure of our amino acid. By the way, next meeting pa naman natin di-discuss yung 3D structure. So pag-usapan na lang natin kung ano itsura niya. So again, sabi natin yung R groups kapag aliphatic, pwedeng hydrogen, pwedeng carbon, and long carbon chains. No? So si glycine, yung R group natin ay hydrogen. No? So that makes it non-polar. Non-polar yung R group natin. So what else, no? Since may dalawa kang hydrogen attached to your alpha carbon, that means this molecule is achiral, no? Achiral yan. So wala siyang chirality center. And this is the only amino acid na walang chirality center. Next one is the alanine. So the R group for alanine is the methyl group. Okay. So yan. For proline, ito yung isa sa mga weird na amino acids. Why? Because kung mapansin nyo, compare nyo yan sa ibang amino acid that is branched out, no? Pag proline, connected yan, no? Connected siya from alpha carbon papunta sa nitrogen. So, naka-pentagon shape yan dito sa gitna, no? So, naka-pentagon shape yan. So, connected yung alpha carbon to the nitrogen terminal group, no? Okay. So, yan yung weird kay proline. Naka-circle siya. For valine, ito yung itsura ng kanyang, ng kanyang R group. So, ako ang trick ko dito kapag valine, itsurang letter V na baliktad yung kanyang R group na. So, naka-letter V yung kanyang R group. For leucine naman, extended lang yan na letter V. Okay? Ito, letter V, extended by one carbon only. For isoleucine, what happens here is that, di ba ito yung leucine mo? You will just move your methyl group to the carbon next to your alpha carbon. Okay, so ito ilipat mo lang dito. So you will have your isoleucine na. And then methionine, ang itsura niyan is ito, may sulfur siya. Okay? So yan. So kailangan nyo ba ito kabisaduhin? Ano na lang, be familiar na lang. Kasi later on, gagawin din naman natin is titignan din natin itong itsura ng amino acids na to. No? Babalik-balikan natin yung itsura niya. Okay. So, yan. Okay, so again, we call this group the nonpolar aliphatic R groups. So, ano meron dito? Yung R group natin is primarily hydrocarbons lang, no? So, hydrogen, carbon, and then may sulfur, no? Si methionine. So, we have 7 nonpolar aliphatic amino acids. Okay. So, ano sila? Ang glycine, alanine, proline, valine, leucine, isoleucine, and methionine. So, 7 yan. Okay, so the next one is the polar uncharged R group. No? So we have 5 of this. No? So sinabi natin polar yung R group because the bond there is polar. No? Okay, and uncharged siya because wala tayong charges sa R group. No? So meaning dipole lang yung mga nasa R group natin. So we have 5 polar R groups. No? 5 polar uncharged R groups na amino acid. Okay? And they are soluble in water mostly. Okay? By the way, kung ito ay water soluble, that means yung nonpolar ay insoluble sa water. No? Okay? So, balik ulit tayo kay polar. So, 5 yan. So, we have the serine, threonine, cysteine, asparagine, and glutamine. Okay? So yan. So for serine, we have the methanol R group, no? So yung methanol attached to your amino acid structure. For threonine, ganun din, although extend mo lang siya ng 1 carbon. For cysteine, no? So instead of having OH at the end, you have your SH group, no? So your SH, your thiol group. Okay. And ito, asparagine. Okay, so asparagine, siya ay amide. So when we say amide, we have the C double bond O NH2. So amide tawag natin doon. And for glutamine, that also consists of another amide group. However, that is 1 carbon longer than asparagine. Okay. So medyo similar tong dalawa, asparagine and glutamine. Okay. So, ano yung pinagkaiba lang nila? Glutamine is longer by 1 carbon unit sa R group. Okay. So, yan. So, mapapansin natin, the ends of the R group are polar, no? Because of the OH, the SH, the C double bond O, NH2 groups, no? Doon. So, hahalo talaga yan sa tubig. So, it can perform hydrophilic interactions. So, pwede yan humalo sa water. Okay. So again, hydroxyl group for serine, threonine. For cysteine, that's a thiol group. And for asparagine and glutamine, you have your amides, no? Your C double bond O NH3. So ilang amino acid na tayo? 7 plus 5, 12 na, no? So let's talk about the next group, the aromatic R group, no? So for the aromatic R group, we have aromatic rings, no? So when you say aromatic rings, ito yung last topic natin sa org chem nun. Pag aromatic ring, basically may benzene ring ka lang, okay? So we have three types of aromatic R groups. So we have the phenylalanine. Okay. So this is phenyl. Okay. This is the phenyl group. So we call this the phenylalanine. We have the tyrosine here. So the difference between tyrosine and phenylalanine is that it contains OH at the end of the benzene ring. Okay. So yun lang yung minor difference nila. So phenylalanine, ayan. Tyrosine, dagdagan mo lang ng OH dito sa dulo. And then we have the tryptophan. So what's weird with tryptophan is that it contains an indole ring. So when we say indole ring sa OrgChem, ito yung itsura nun. So may benzene ka na may kadugtong na 5-membered ring. 5-membered ring. So 5 carbons yung nasa ring na kadugtong niya. So this is called the indole ring. Okay. So pero overall, ang malalaman natin kapag aromatic group is that meron siyang benzene rings. Okay? May mga benzene rings sila. So yan, so we have the 3 aromatic R groups, phenylalanine, tyrosine, and tryptophan. Okay lang naman kahit di nyo makabisado. No? So to be honest with you, nakalimutan ko na rin yan slightly kasi 1 year na since last ko to naturo. No? Okay, pero naalala ko pa yung most na structure. Okay. Pero sa case nyo ngayon, online class naman tayo. So, will be flexible. No? So, pwede nyo tignan-tignan yung structure. Kasi in the end, ang important lang naman dito is alam nyo yung ano yung pwede nyo gawin, no? Not the structure. Okay. So, next one, we have the positively charged basic R groups. No? So, when you say positively charged R group, may charges sa ating R group. No? Okay. Basic in a sense that it contains nitrogen atoms that are charged. No? So nitrogen usually behaves as a base, di ba? Especially if it is in ammonical form. So ibig sabihin naka-ammonia siya. Okay. So we have the lysine, the arginine, and the histidine. Okay. So ito yung lysine. Okay, so we have the 4 carbon chain here, and at the end you have your amine group, your NH3 group. Yung arginine, ito naman yung itsura niya. Kung mapapansin niyo yung arginine sa dulo niya, meron siyang cluster ng carbon connected to 3 different nitrogen groups. No? So ang tawag natin dyan ay guadino group. No? Guadino group tawag dyan. Okay, so whenever your carbon is connected to 3 different nitrogen atoms, tawag doon ay guadino. No? Okay, and then for histidine, we have the thing called imidazole. So that is another 5-member ring. However, may nitrogen dyan. No? That is different with the indole ring here. No? So yung indole ring here kasi is meron kang benzene and the 5-membered ring. For imidazole, you just have the 5-membered ring. No? So yeah. So all of which no, ay positively charged ang R group. No? So they can be ionized. No? Yung mga nitrogen connected to hydrogen, yun yung mga pwede i-ionize. No? So ito, pwede ito maging positive charge. Ito, and this one. Ito, side kwento lang na yung urea from our urine no, ay galing sa production ng arginine. No? So itong arginine na yan, pag nagamit yan ng ating katawan, byproduct niyan is the urea. Okay. So ito yung urea part. No? Okay, so ayan yan. So ano ba itsura kasi ng urea? Ganito lang yan. No? C double bond O, NH2. NH2. Diba? Nagre-resemble siya dito sa guadino group. No? So yan. Dito nanggagaling yung urea sa ating ihi. No? And lastly, we have the negatively charged acidic R group. No? So we have the two, aspartate and glutamate. No? Don't confuse yourselves with asparagine and glutamine. No? Kasi aspartate, glutamate. No? So isipin nyo kapag 8, ano meaning ng 8 sa OrgChem? Carboxylic group yan. Okay? So aspartate and glutamate, they both have carboxylic group, and just like the asparagine and glutamine, the glutamate is one carbon longer than the aspartate. Okay? So yun yung subtle difference nila. Okay? So again, asparagine, instead of having the carboxylic group here, you will have the amide group, no, the C double bond NH2, C double bond O NH2 or NH3. Pag glutamate, ganun din. Glutamine, C double bond O NH2. However, if yan ay aspartate and glutamate, no, instead of having the amide group, you have your carboxylic groups, no. Okay, so yan. So again, mabilisan na tingin tayo. Although pwede nyo naman to balik-balikan, no? Kasi hindi naman natin kailangan kabisaduhin to. Okay, so we have five different groups for our amino acids. We have the nonpolar aliphatic R groups. So 7 yan. We have the glycine, the alanine, the proline, the valine, lysine, isoleucine, and methionine. Okay. So ano yung R groups nila? Primarily primarily aliphatic groups lang, such as hydrogen, carbons, no? So yan. Okay, so by the way, since ito ay non-polar, that means hindi yan magpa-perform ng hydrophilic interaction. Pwede yan mag-hydrophobic interaction. So polar uncharged R groups. So that means polar yung ating R group. So tatlong characteristic yan. Pwedeng hydroxyl group, pwedeng sulfohydryl or thiol group, o kaya pwede ring amide group, no? Okay, so yun yung tatlong characteristic, no? For the hydroxyl group, you have the serine and threonine, pareha sila may OH. For your sulfohydryl group or thiol group, you have your cysteine. Cysteine basa nila dyan, no? And then for amide groups, you have the asparagine and glutamine. Okay, so again, di ba mapapansin yung aspartate and glutamate, similar siya with the two. Pag itong NH2, pinalitan mo ng OH, no? Ano yun? Aspartic acid, no? Aspartate. Pag ito, pinalitan mo to ng OH, itong NH2, pag pinalitan mo ng OH yan, that will be glutamate, no? Or glutamic acid. So, yun yung mga subtle difference lang. And then we have the aromatic R groups, no? So when we say aromatic, ibig sabihin yan may aromatic ring or mayroong benzene ring. So tatlo lang yan. We have the phenylalanine, tyrosine, and tryptophan. No? So phenylalanine and tyrosine, they are similar. They both have the OH, okay? They both have the OH. No? Ay, yung isa may OH, yung isa wala. Okay. And then we have the tryptophan. So the tryptophan is an extended, that is an extended benzene ring kasi meron siyang 5-membered ring sa gilid niya, no? So this group in particular, we call this the indole ring. So next group will be the positively charged R groups. So yan, we have the lysine, arginine, and histidine. So kapag positively charged R groups, amine yung nasa dulo nila. Okay, so yan. And ano yung mga key ideas natin dito? Yung arginine, ito yung nagiging source ng urea sa ating urine after the protein is consumed by the body. Okay, so yan, maglalabas yan ng urea from the guanidino group. No? And then ito, imidazole ring, this is similar with the indole ring, however, wala siyang benzene na kasama. Then last one, we have the negatively charged R groups. So dalawa lang yan. You have the aspartate and the glutamate. No? So Compare that to asparagine and glutamine. Aspartate and glutamate have carboxylic ends instead of the amide ends. No? Okay. So to further help us sa pag-memorize ng mga amino acid, we sometimes use the 3-letter abbreviation and the 1-letter equivalent of which. So for every amino acid, pwede natin siya i-represent as 3 letters or pwedeng 1 letter lang. So for example, alanine can be abbreviated as ALA or ALA. And the single letter abbreviation for that is A. For arginine, that's ARG. And this 1-letter abbreviation is R. Okay. For asparagine, the abbreviation is ASN, or the 1-letter abbreviation is N, and so forth, and so on and so forth. So do you have to remember this? Kahit di na, no? I'll give you the freedom naman to use this slide whenever you are studying. So kunwari, you have an amino acid chain composed of Chester. So kunwari, yan yung 1-letter abbreviation. So we have an amino acid chain composed of Chester. What are the amino acids involved? So ano yung letter C? So letter C represent cysteine. Yung H that represents histidine. Yung E that represents glutamine, glutamic acid or glutamate. Glutamic acid and glutamate are the same, ha? O yung S, ano yung nire-represent niyan? Serine. Mamaya gagamitin natin to. Yung T, it represents threonine. So may OH yan, alcohol yan. E, another glutamate. And the R, you have your arginine. Okay, so yan yung ways namin to abbreviate the amino acid sequence. Ito rin yung ginagawa ng mga Ayan, nag-charge ako. Yan din yung ginagamit ng mga nagsisequence ng viral protein ng COVID-19. So kung makikita nyo, sinasabi nila D614G, yung D doon, ibig sabihin nun aspartic acid. Yung G, meaning nun glycine. Okay. So yung mga letters na yon, may katumbas yun na amino acid. So malalaman natin kung ano yung effect kapag napalitan yung amino acid sa protein next week na. So yun lang. Kunwari sa sickle cell, ano yon, from L naging V, no? So ang nangyari doon is from lysine naging valine. Napalitan yung amino acid na. So yun yung mga shorthand notation namin to identify the amino acids na pinapatukuyan. Okay? So again, be familiar na lang with this one kahit di nyo nakabisaduhin. Okay. So these are some of the uncommon amino acids which are found in the body. So ang kanilang function ay variable. So hindi siya fixed to one function only. So for example, we have the proline molecule here. So again, as I told you kanina, ito yung itsura ng proline. Yung alpha carbon, connected yung R group niya sa nitrogen end. So proline yan. If you attach OH on the second carbon ng R group, you will have your hydroxyproline. Okay? Nakikita yan sa katawan, although hindi siya abundant, kasi function niya ay iba-iba yung functions niya. So, ganun din yung lysine natin. Pwedeng lagyan natin ng OH yan. So, we will have hydroxylysine. Tyrosine can be extended to produce thyroxine. Okay. So, yan. So, yan yung mga uncommon amino acids that are found in nature. And mas marami pa dyan. Marami-rami talaga yan. But for the purpose of our class, only the common amino acids has to be memorized or has to be familiarized. Okay, so again, nonpolar amino acid, ang characteristic nila ay hydrophobic because they are nonpolar, so they will not dissolve in water. When we say polar amino acid, these are the amino acids that will dissolve in water because they contain polar groups or they contain charged particles. No? So pwede sila mag-form ng ion dipole interaction with the water. And ito yung ating amino acids that cannot be made by the body. So that is WHT, MILK, FVR. No? Ang tawag namin dito ay what milk favor, no? Pwede rin naman William Howard Taft Milk Ferdinand Fidel V. Ramos. No? So yan yung mga tawag namin dito. William Howard Taft Milk Fidel V. Ramos. No? Kaya what milk favor, no? So ano-ano amino acids yung essential, ibig sabihin yung kailangan natin kunin sa diet, no? So W, that's tryptophan, H is histidine, T is threonine, M is methionine, I is isoleucine, L is leucine, K is lysine, F is phenylalanine, okay? V is valine, and R is arginine. No? So yung mga amino acids na yon, sila yung kailangan kunin sa diet. No? Okay. So yung mga aromatic amino acids natin, nakukuha natin yon mostly sa halaman. No? Yung colors kasi ng halaman, that can be attributed by the presence of benzene rings doon. No? So yun. So that's why we need gulay din. No? Source din sila ng amino acid other than other other dietary products, no, such as lean meat, no, white meat, and and ano pa ba yung source nun? Ay mga gulay, etc. No. So yan, dun sila nakukuha. So again, yung mga may benzene ring, mga aromatic na amino acid, most likely nakukuha sila sa gulay. Okay, so again, what are the essential amino acid? William Howard Taft Milk Fidel V. Ramos, no? WHT, MILK, FVR. No? So ano-ano uli yan? So that's tryptophan, histidine, tyrosine, threonine, um, ano uli yung M? Methionine, isoleucine, leucine, K is lysine, F phenylalanine, valine, and arginine. So yan. So yan. So sila yung mga essential amino acids. Okay? So yan. So once we have our amino acids in the body, they can serve as either base or an acid, no? So anong nangyayari kasi, we have the functional groups for amino acids, 'di ba? So yung functional groups na yon, they can be ionized, no? Anong particular functional group yung na-ionize sa ating amino acid? So number one will be the carboxylic groups. The other one will be the amine group. And the last one will be the R group, no? So yung tatlong groups na yon, they can be ionized, no? So they can be protonated or deprotonated at different pH, no? So for example, at very acidic pH, ibig sabihin nun, protonated lahat ng R groups, no? So when you say protonated, may hydrogens, no? Kapag mataas naman yung pH, meaning basic nun, deprotonated yung R groups natin. Okay, so mababawasan sila ng hydrogens na. Okay? So whenever we have an amino acid that is electrically neutral, so when you say electrically neutral, zero yung overall charge niya, we call that the Zwitter Ion. Okay? So for example, we have this amino acid here wherein R group is not indicated. So at acidic media, the carboxyl group is protonated. The amine group is also protonated, no? Whenever your amine group is protonated, yung NH2 nagiging NH3+, no? So let's determine ano yung charge ng molecule na to. So we have the positive one charge and other groups do not have any charges. So that means the overall charge for the acidic amino acid is positive 1. However, when it starts to dissociate, no, whenever our amino acid starts to dissociate its proton, ano mangyayari? For example, ang first na madi-dissociate ay yung sa carboxyl group. So, mawawala yung H. What will happen is that yung matitira sa kanya, magiging negative charge yun. Di ba itong CO2H, matatanggalan ng H. So, matitira doon, carboxylate ion na lang, COO minus. Okay. So, if you get the overall charge of this molecule, we have positive 1 for the amine group, negative for the carboxylate group. So, plus 1 minus 1 is 0 charge. No? So, when our amino acid is in neutral form or 0 yung kanyang net charge, that is called the Zwitter Ion. And then, as you increase the pH higher, what will happen is that the amine group will lose its proton kasi mataas na yung pH, no? Mababa na yung concentration ng H+. So, mangyari, yung ating H dito sa amine group, mawawala yan. So, as a result, mawawala yung positive charge kay nitrogen. So, if you get the overall charge for this molecule, that will be negative 1. Okay. So we can use this concept of Zwitter ion in order for us to obtain proteins, no, from any products or any dairy products, no. So kunwari, you want to get the casein from the milk, no, pwede mo yung gawin. You just need to adjust the pH of your milk, no, in order for the casein to precipitate, no. So adjust mo yung pH until the entire casein molecule is a Zwitter ion, no. So once na naging Zwitter ion yon, no charge na yan, magiging nonpolar yung buong molecule na yon. So they will start clumping, no. That's why yung gatas kapag napapanis, nagka-clump, no, because it turns into a Zwitter ion. Yung mga proteins doon, naging Zwitter ion. So nagka-clump sila because they don't have charge, no, overall. Okay? So when we say isoelectric point, no, so isoelectric point pertains to the pH in which your amino acid has zero net charge. Ibig sabihin nun, dun siya nagiging Zwitter ion at the isoelectric point. No? To solve for the isoelectric point, that is Pi, okay? Hindi yan mura, no? Pi talaga tawag namin dyan. So for isoelectric point, that's Pi equals the pK1, the pK2 divided by 2. No? So ano yung pK1, pK2, no? So pK1 and pK2s are the they are the dissociation pH, no, ng ating acid, no. So at those pH doon siya mag-start mag-dissociate, no. Let's take a look at this example. So this example is glycine. Okay, so glycine itong molecule na to. At low pH, ammonia is protonated. The carboxyl group is protonated. The R group wala siyang pake kasi hindi siya reactive with hydrogen kasi hydrogen na siya, no. So yan, at low pH, glycine has an overall charge of positive 1. Okay? However, as your pH starts to reach pK1, pK1 is the point at which your carboxyl group will start dissociating into ions, no. So yan, pag na-reach na natin yung pH equivalent to pK1, okay, so dun na mawawala yung H ng carboxyl group, no. So, magiging COO minus na lang siya. Magiging acetate group na lang siya. So kapag kukunin mo yung net charge ng molecule niya yan, that is 0. So we call it the Zwitter ion. So that is also the isoelectric point. That is the pH at which your molecule is Zwitter ion or wala siyang charge. However, if you further increase the pH, no, you will arrive with the pK2. Okay, so the pK2 is the point at which your amine group will start losing electrons. No? Losing protons. And as it loses electrons, mawawala yung positive formal charge kay nitrogen. So instead of having NH3+, you will have NH2. So si carboxylate na lang yung may charge, no, for glycine. And the overall charge of the molecule here will be -1. So it is now an ion. So if you want to determine the exact Zwitter Ion pH, basically, hanapin mo yung pK1, pK2, then i-divide mo lang siya by 2. Okay. However, itong example natin, hindi natin kino-consider yung R groups na R groups na ionizable, yung positive R groups and yung negative R groups, no? What if tatlo yung pK values mo? Madidissociate yung COOH, madidissociate yung NH3, and madidissociate yung R group. In such cases, kapag tatlo na yung magdidissociate, in order for you to get the PI or the isoelectric point of your molecule, of your amino acid, ganito lang gagawin nyo. Tignan nyo yung tatlong pK values. No? Kapag may dalawang lowest doon, yun yung i-average mo. So you will get the PI of the molecule. Kapag dun sa tatlong pK values, may dalawang malalaki yung pH, no? Ibig sabihin on basic yun yung kukunan mo ng PI, no? So yun yung i-average mo to get the PI. No? So let's have some examples here. So we have the pKa table here. Okay, so ito gagamitin talaga natin to. We have the pKa values of different amino acids. So let's try to get their PIs, no? Their isoelectric point. Huwag kayong ma-awkward sa PI ha. Isoelectric point tawag doon. Okay, so let's have alanine. So alanine, ano itsura ng alanine na yan? So let me draw that. Upsi, mali ang aking plus. Ulit-ulit tayo from the top. Okay, so we have here alanine. So at very high pH, okay, at very low pH pala, sorry. So at very low pH, our amine group is protonated and our carboxyl group is protonated. No? The R group is unaffected kasi nonpolar yan. So hindi yan magre-react with acids. Okay. So, okay. So ano yung gagawin natin? So let's start deprotonating the molecule na. So looking at the pK table, tingin tayo sa pK table na maigi. Ano may lowest value? Ang may lowest value is the pK for the COOH group. No? So that means if we reach the pH equal to 2.34, doon yan magkakaroon ng, doon mag-start yung dissociation ng carboxyl group. No? So again, so suppose nag-start tayo with pH equal 1 dito. As we start approaching pH equal to 2.34, what happens is that your carboxyl group will start dissociating. Okay? So, anong mangyayari doon? So, yung carboxyl group mo, instead of being COOH, magiging COO minus na lang siya. Okay? So, at that pH, dun siya mag-i-start ma-dissociate. Okay? Then, after that, no, as we reach pH equal to, ano yung susunod na pH? Ito, 9.69. So at pH starting 9.69, what happens is that yung ating amine group, dun siya mag-start ma-dissociate. No? So itong NH3 plus, magiging NH2 na lang yan. No? So mawawala yung isang hydrogen niya. So let's determine the overall charge for each molecule here. So this one, that is positive 1, walang charge, walang charge, walang charge. So overall, that's positive 1 in charge. This one, alamin natin yung overall charge. Positive 1, negative 1, 0, 0. Plus 1 minus 1 is 0. So this is the Zwitter Ion version of our molecule. However, itong last na dissociation, alamin natin yung overall charge. That's negative 1, 0, 0, 0. So Negative 1 plus zeros is negative 1. Okay. So if we are asked, no, what is the pH at which your amino acid is in its full Zwitter ion form? Ibig sabihin, completely Zwitter ion siya. Anong pH? Exactly yon. So, gagawin natin is just to get the average of the two pHs, no? So, yung nasa gitna nun, that will be our isoelectric point. Doon yung pH kung saan fully Zwitter ion ang version ng ating amino acid, no? So, basically, itong dalawang pH na to, i-average mo lang para malaman mo at which pH siya fully Zwitter ion form, no? So that will be 2.34 plus 9.69 divided by 2 para makuha natin yung PI, no? So add lang natin. So at pH 6.02, ibig sabihin, at that pH, dun Zwitter ion yung ating molecule. Okay, so dun siya magiging Zwitter ion. So let's try another one. Let's try arginine. Let me draw arginine lang. Medyo malaki tong molecule na to. Okay. So arginine, this is ito yung may tawag doon, guanido group, no? So I will draw the protonated form of the arginine. So ibig sabihin ng protonated form, yung carboxyl group may hydrogen, yung amine group sobra ng hydrogen. And then yung R group ay protonated din. Okay. CH2, CH2, CH2. Then ito na yung guanido group. Okay. So this is the ang haba pala ng drawing ko. So this is the form of arginine at lowest pH, no? Ibig sabihin at acidic medium, all the ionizable groups are protonated. The COOH, the NH3+, and the N of the guanidino group, no? So, protonated sila lahat, no? Baka ito question, sir, paano namin malalaman kung saan yung protonated? Tingin lang kayo dun sa drawing ng amino acids na binigay ko kanina. Okay. So, dun yung malalaman kung saan yung protonated or hindi, no? Kung saan siya mapoprotonate or not, no? Pag yan, kapag yan ay positively charged amino acid, most likely, yung nitrogen yung mapoprotonate. Okay. So yan, so this is our arginine at the lowest pH. So tingin tayo dito sa table. So as we reach pH 2.17, what happens? Yung iyong carboxylic group, it will start dissociating into ions, no? So dun siya mag-start deprotonate, no? So at pH 2.17, okay? So palitan natin. So at pH 2.17, it will start dissociating. The drawing na naman ako. Wait lang. Okay, so madi-dissociate na yung ating R group doon. Yung ating carboxyl group na. Okay. CH2 NH. Mas madali to gawin pag nasa whiteboard eh. Pero ayan. Okay. So at pH 2.17, your carboxyl group will start dissociating. Ano susunod na ma-dissociate? Tingin tayo dito sa pK values. So tapos na tayo sa 2.17, ang susunod ay 9.07. Okay? So at pH equal to 9.07, what will happen? Your NH3 will start dissociating. Okay. So, sabi doon, yung NH3, doon siya mag-start ma-dissociate. No? Okay. So, let's do that. No? Yung NH3 na. Wait lang. So, maging H2N na lang yan. No more charge. COO minus pa rin yan. CH2, 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 NH, C double bond C and nagkamali. Wait lang. Burahin natin. Yan. Oops, alo. Naburo lahat. Ano ba yan? Ulit na naman. Wait lang ha. Let me redo that. COO minus. CH2, CH2, H2, NH, C double bond NH2 plus NH. No? So again, at pH 9.07, our ion, no, our amine group, no, yung NH2 group, that will start dissociating into ions. So what happens, yung NH3 plus mo magiging NH2 na lang. Okay, so wala na siyang charge. Wala nang formal charge yung nitrogen. And then lastly, at higher pH, so at pH 12.48, sabi dito, at pH 12.48, your R group will start dissociating. So let us visualize that. No? Pakita natin. Nakalimutan ako ng pH yan. 12.48. Okay, so at pH 12.48, your R group will start dissociating. Ang tanong, saan dito sa R group? It is dun sa may nitrogen. Okay, so usually naman nitrogen yung nagdi-dissociate na. CH2, 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 NH, C double bond N. Yan, madi-dissociate yung H dyan. So instead of having NH2 plus, that will now become NH na lang. Okay? So this is the process of dissociation ng protons sa ating arginine. Okay? So yan yung process ng separation ng H plus, no, from our molecules. So we always start with the carboxyl group, no? Again, we always start with the carboxyl group. Then yung susunod doon, it's either the N group or the R group na, no? So, yan, variable na yun. Just look at this table na lang para malaman nyo kung ano yung sequence ng dissociation nila. Okay. So, yan. So, after writing this, no, na-track down na natin yung changes sa groups ng ating amino acid, let's determine the overall charge. No? So let's determine the overall charge starting from the leftmost side. Bilangin lang yung charge particles. So dito, positive 1, yung COOH, wala sang charge. So, positive 1, positive 1, positive 2. Okay, so once our carboxyl group starts dissociating, what will happen? Magkaka-negative charge na dun. So from positive 2, ano na magiging charge? Positive 1, negative 1. So overall, that's positive 1. Then what happens next, no? At pH 907, your NH group, no, or your NH3 group, no, will start dissociating, no? So mawawala na yung positive formal charge for nitrogen. So matitira na lang is positive 1 and negative 1. So this is our Zwitter Ion, no? So 0 na yung charge ng molecule na yan. And then let's try the last one. So for the last one, we have this molecule here. In last dissociation would be the R group. Yung nitrogen doon, magre-release siya ng hydrogen. So mawala yung formal charge kay nitrogen. No? So ano yung overall charge ng molecule natin? At pH beyond 12.48, that will be -1 in charge. No? So if you want to precipitate your arginine, at what pH dapat siya ilagay para mag-precipitate yan, para mamuo yan? Okay. So alamin natin. Paano natin malalaman using PI, no? So let's use the PI to determine kung saan siya magiging Zwitter Ion. And that's PI equals yung in between, no? Okay. So tignan nyo, yung dalawang highest, yun yung basis natin for PI kasi nandun sa gitna niya yung Zwitter Ion. Okay. So yung dalawang highest yung pagbabasihan kapag basic amino acid yan. Doon kasi natin makikita yung Zwitter Ion form sa gitna ng dalawang pK values na yan. So kailan yan magpe-precipitate? Okay, kailan maximum yung precipitation? So kunin mo lang yung average ng dalawang pH. So that will be 10.78, no? At pH equal to 10.78, that is the pH at which your arginine, no, can be precipitated at its maximum amount, no? So doon natin sila makukuha lahat-lahat talaga, no? Okay. Let's try some pa, no? Kuha pa tayo ng iba pa. Okay, last one. So pwede gawin nyo mamaya after the class, no? And since may holiday naman next week, no? Kunwari, sinipag lang kayo, pwede i-try nyo lahat yan. Kasi nung ako nag-aaral nito, ginawa namin to lahat eh. Nakakatamad, no? Pero ang punto lang doon is that kapag gusto mo kunin yung PI, kung saan magiging Zwitter Ion yung iyong molecule, tignan mo lang yung dalawang mababa or yung dalawang mataas na pH, no? Pero kung dalawa lang yan, i-average mo na lang sila. Pag tatlo, piliin mo yung dalawang mababa or yung dalawang mataas. Doon mo makikita yung Zwitter Ion. So, pwede nyo gawin yan, no? After the class. So, let's try some more. Yung acidic naman tayo, let's have aspartic acid, no? Aspartic acid. Hindi yan yung ascorbic acid, ha? So let's draw the aspartic acid at its lowest pH. That means N group, the carboxyl group, and the R group are all protonated now. Okay, so this is our aspartate, aspartic acid. And so CH2, C double bond O, OH. If I extend this by one carbon here, that will be glutamate or glutamic acid. Okay. Ayan. Ano susunod? Start na natin siya i-dissociate. Tingin tayo sa pK value. Sino yung unang matatanggal? We have the aspartic acid here. So, unang matatanggal yung pinakamababa, which is the carboxyl group na. So, sabi ko nga sa inyo, yung carboxyl group, yun yung unang natatanggal parate. No? Okay. So, let's do that. No? So, at pH, ano yun? 2.09. So, at pH 2.09, your carboxyl group will start dissociating. No? So, let me draw that. So, instead of having COOH sa carboxyl group sa taas, wala na siya. So, that's pH 2.09. That's the pK of the alpha carboxylic group. Ano susunod? Tingin tayo sa pK table. After 2.09, susunod will be 3.86 na. Side chain yung diprotonate or yung R group. So at 3.86, yung R group yung mawawalan ng H na. 3 point, ano yun? 3.86. So at pH 3.86, okay, your R group will start dissociating. Ito question, Sir, saan dyan? Doon sa may COOH. No? Again, kapag yan ay basic amino acid, yung nitrogen yung matatanggalan or matadagdagan ng proton. Pag yan ay acidic, carboxylic, kapag yan ay acidic na amino acid, yung COOH yung magbabago. Pwedeng madagdagan, pwedeng mawalan ng hydrogen. Okay, so in our case, as we reach pH 3.86, mawala na ng H yung ating aspartate. C double bond O, O minus. And then lastly, yung N group na yung matatanggalan. So tingin tayo, N group. Okay, so the N group will start dissociating at pH 9.82. 9.82. So instead of having NH3, maging H2N na lang yan. Okay. So that is our sequence ng dissociation. So just look kung sino yung pinakamababang PK. Tapos yun yung mauunang madidissociate. No? And then sequence na lang yan. No? Pwedeng yung R group mauna or R group yung susunod. Pwedeng amine group yung susunod. No? So just look at the table. So let's check the overall charge of the molecule at different pHs. No? On the leftmost side, we have one charge only, positive 1. So the molecule here is positive 1 in charge. No? The next molecule here, we have positive 1 charge and negative 1 charge. So basically, this is the Zwitter Ion. Okay? And as you can see, pag acidic yung inyong amino acid, ang Zwitter Ion ay makikita in between the two lowest pHs. Okay? Ayun, dalawang lowest na pH. Then, sunod tayo. After pH 3.86, ano yung magiging charge ng ating ion? This will be negative 1 na, no? So, positive 1, negative 1, negative 1. Overall, that's negative 1. And then lastly, we have negative 2 charge here. Okay. So, if you want to precipitate, no? If you want to precipitate out your aspartic acid, kung gusto mo siya mamuo, anong pH dapat? So the pH is equal to the PI, no? So let's solve for the PI. Yun yung pH na nasa gitna, yung Zwitter ion. Okay, so that will be 2.09, 3.86 divided by 2. 2.09, 3.86 divided by 2. So that will be 2.98. So at pH 2.98, doon tayo makakita ng Zwitter ion form of our aspartic acid. No? So yun yung Zwitter ion form niya. Okay? You can do that for the rest of the items here. No? Pwede nyo itry yan. No? Okay, so sa quiz natin, most likely, ganun din yung mangyayari. No? So question doon, kailan mo makikita ito as Zwitter ion? Basically, pwede mo i-drawing yung lahat or kaya pwede mo i-average na lang yung dalawang lowest or dalawang highest. No? Okay, so ganun lang. Okay, so that's it. No? So yan. So aning makukuha natin idea dito? That amino acids can both act as acid and base. No? Acid because it can accept protons, no, at lower pH, and bases because it can acid kasi nagre-release yan ng protons at lower pH. No? Bases kapag yan ay tumatanggap ng protons. No? So yan. Ano nga pala tayo? Bronsted definition. Pag Bronsted definition, pag nagbibigay ng H, acid. Pag tumatanggap ng H, base. No? And we just saw that our amino acid can do that. No? Pwede siya tumanggap or mag-release ng H. No? So yan. So acids can really act as acids and bases. No? To get the pH at which your amino acid is a Zwitter ion, meaning wala siyang charge, you just have to get the PI or the isoelectric point. No? Ang technique dyan is that if you have acidic amino acid, kunin mo yung PI ng dalawang lowest pK values. Pag basic yan, yung PI mo will be the two highest pKa values. No? Pero kapag yan ay ano lang, uh, hindi siya acidic, hindi siya basic, for example, yung mga aliphatic, just average out yung dalawang pK values. Okay? So, ganun lang. So now let's talk about the peptide bonds. No? So what are peptide bonds? Peptide bonds are the linkages of amino acid, no? As it forms the protein. Okay. So sabi natin, yung amino acid, pag nagdugtong-dugtong yan, it will become a protein, no? So saan nangyayari yan sa cell, no? That happens in the ribosome, no? Remember your mRNA, no? Kung naalala pa sa inyong high school days, no? Okay, yung mRNA nyo, that that gives the ribosomes, no, the code kung anong amino acid yung kailangan gawin in the form of codons, no. 'Di ba yung AUG, UUG, something something. So yon, yung codons na yon that will tell what type of amino acid must be placed there, no, to produce the protein. Okay. So, ang nangyayari doon, kapag pagdudugtungin na ng ribosome yung amino acids, they form the peptide bonds, no? So, what are peptide bonds, no? Peptide bonds are the linkages nga of two amino acid and that is produced when two amino acids undergo dehydration reaction. So, ito yung ating itsura niyan. So, this is our amino acid. So, it will undergo dehydration reaction. So what happens is that the carboxyl group of the amino acid, no, and the amine group of the other amino acid, ang mangyayari doon is magdudugtong sila and water will be eliminated during that process, no. So itong dalawang hydrogen ng amine and yung oxygen ng carboxyl group, mawawala yan. And we will form the peptide bond here. Okay. The peptide bond is an amide bond, no? Amide bond yan. Bakit? So you have your C double bond O and nitrogen group here. So C double bond O, nitrogen group there. So that is a peptide bond, no? Ano pa yung characteristic ng peptide bonds, no? By the way, ito yung tsura ng ating ng ating proteins pag nagdugtong-dugtong na yung amino acid. So you have this amino acid, the N-terminal is on the leftmost side, okay? So ito yung carboxyl group, ito yung N group ng other amino acid, magdudugtong sila to form the peptide bond, no? Okay? So yan, continuous yan. So ito yung new amino acid natin at the carboxyl group that will connect to another nitrogen group of the other amino acid to form the peptide bond, and so on and so forth, no? Okay? So yan. So by the way, when drawing the peptide bond, the N-terminal is always on the leftmost side and on the rightmost side, you have your C-terminal there, no? Okay, so ganun. Anong characteristic ng peptide bond, no? So the characteristic of the peptide bond is that it can undergo resonance stabilization, no? Because meron tayong delocalized electron doon. So ORChem ulit to. So ano ba yung tsura ng peptide bond, no? So ganito yan, you have the C double bond O, nitrogen, and then kadugtong niyan yung iba, no? Okay, so yan. Yan yung itsura niya. Since the electrons here are delocalized, kung naalala nyo pa yung criteria ng delocalization natin, lone pair, then pi bond, no? Pwede yung mag-delocalize, pwede ito mapunta dito, so ito magiging lone pair, di ba? So that means the peptide bond is resonating, no? And because of the resonance of the peptide bond, we have the partial double bond character of this bond, making it stronger than the normal single bond, no? Bakit naging partial double bond character, no? Kasi pag resonance, ang bond order nun ay ano, 1.5, no? 1.5 yung bond order niya. Ibig sabihin niyan, parang average siya ng ano, dalawang bonds tas isang bonds, no? Pag kinuha mo yung average doon, 1.5. So partial double bond character. Okay, so again, paano ulit naging partial double bond character? Pwedeng single bond siya, pwedeng double bond. Pag in-average mo yon 1.5, no? Or partial double bond character. So that's why peptide bonds are stronger. And luckily for us, ganun yung case, no? Kasi imagine if your peptide bond is weak, what will happen to your proteins? It will be easily denatured, no? Mabilis masisira yung protein mo. Hindi tayo mag-e-exist. Imagine nyo yung unang panahon, di ba? The earth is very hot, no? So imagine yung super init na mundo, yung heat nun, pwede nun sirain yung peptide bonds, edi walang life na nag-form, no? But because of the partial double bond character of the peptide bond, because of its resonance, mas matibay siya, no, compared to other single bonds, no. Also, our peptide bonds are planar. So, ibig sabihin flat yan. Bakit, no? Because the hybridization of our atoms here is somehow sp2 ang itsura nun. sp2 hybridized na. Kasi you have the double bond here, sp2 yan. Pag napunta yung double bond dito, ito maging sp2. So ang average na geometry ng dalawang atoms na to, nitong amide groups natin sa peptide bond is sp2 hybridized or trigonal planar. So ibig sabihin flat itong part ng molecule na to. Okay? So imagine that as ito ay flat na triangle. Ito din, flat na triangle yan. Flat na triangle. Flat na triangle. Yan yung mga trigonal planar sa ating amino acid chain. However, the other atoms there ay hindi na trigonal planar yan. Ano na yan? Ano na sila? Tetrahedral na sila. That's why may 3D shape pa rin yung protein natin. Only the peptide bond is planar because of the sp2 hybridization of the atom when it undergoes resonance stabilization. Okay? So, yan. So, practice tayo. From this molecule, let us identify the amino acids, no, and the peptide bonds here. Practice tayo. So, again, when drawing the protein on the leftmost side, you have your amine groups. On the rightmost side, you have your carboxyl groups. No? Ganun yung sequence when our proteins are made in the ribosomes. Okay, so let's do that. Paano tayo mag-start dito? Start ka sa leftmost side. You have the nitrogen, the alpha carbon, the alpha carboxyl group. No? Once you have the alpha carboxyl group, yun yung start ng peptide bond. Okay? So again, paano uli? Nitrogen, alpha carbon, alpha carboxyl group. Then pag nakita mo yung double bond may katabing H. Kapag yung double bond O may katabing N, amide na yun. No? So that means ito ay amide. Ito, si double bond O may katabing N. This one is also amide. Peptide bond, no? To be exact. Okay, so ito double bond ON. So this one is another peptide bond. No? This one is another peptide bond. Okay. Okay, so yan. So yan yung peptide bonds natin. How many peptide bonds do we have, no? So since meron tayong apat na double bond ON na motif, no? Ito double bond ON, double bond ON, double bond ON, double bond ON. So since may apat tayong ganyan, apat na peptide bonds meron tayo dyan. So pag tinanong sa inyo, ilang amino acid meron dyan, ganito lang. +1 lang kung gaano karami yung peptide bonds. No? So meron tayong 5 amino acids dyan. So ano-ano yung mga yon? Ito yon. So you have this, you have this, you have this, you have this, and you have this. Let's identify the sequence of the amino acid. Identify natin sila. So this is a benzene ring with OH. Ano yan? Kung naalala pa, yan ay tyrosine. No? Tingnan mo, napaisip din ako. Tyrosine nga ba? Phenylalanine, tyrosine. Okay, tyrosine. Nasa isip ko yung threonine, pero yung threonine may OH nga pala. No? Okay. So that is tyrosine. Paisip pa talaga ako nun. How about this one? How about this amino acid? Medyo hindi obvious, no? Teka lang. Imaginein natin. This is the N-terminal. This is the carbon, alpha carbon. This is the carboxyl group. So ito yung R group. Ito naka-point palabas. So let us visualize the R group. So may CH3, CH, and OH, no? So this is polar amino polar R group 'yan. So meron kang CH3, CH2, and OH, no? So ano 'yan? Ito yung threonine, okay? So ito yung threonine. Okay? So yan. Tignan nyo na lang yung PowerPoint ulit, no? Para may reference kayo. CH3, CH, OH. Threonine yan. Okay. How about this one? Well, we have CH2, CH2, and the amide. Ano to? Ito ay glutamine. Glutamine. Okay. So ano difference niya with the asparagine. Ang asparagine, isa lang yung CH2. Ang glutamine, dalawa yung CH2 followed by the amide group there. Okay? What's the next amino acid? Itong nasa taas. Itong nasa taas, this is, makikita nyo, yan ay may indole ring. This is tryptophan. Okay, so that is our tryptophan. Okay. Then, the last amino acid here, mapapansin natin dyan sa last amino acid na yan, it contains carbon groups lang. So, that is Aliphatic R Group, no? So, pito yung ating Aliphatic R Group. Alamin mo lang kung ano yung format niya. So, CH3, CH2. Ito yung alpha carbon, CH3, tas may dalawa dito sa kabila. So, that will be Isoleucine. Okay. So, Isoleucine yung itsura niyan. Okay. Again, you can use the PowerPoint now when identifying anong amino acid yon. Okay. So, yan. So we have 4 peptide bonds and we have 5 amino acid for this specific protein, for this specific amino acid sequence. No? So let's try to write the 3-letter abbreviation for this. Ano yung 3-letter abbreviation niya? Tyrosine muna. So tyrosine will be TYR followed by the threonine. Okay. Threonine is THR followed by the glutamine, which is GLN, followed by the tryptophan. Tryptophan is TRP, followed by the isoleucine, which is ILE. Okay. So this is the 3-letter sequence for our amino acid. No? However, for us to indicate clearly kung sino yung nasa N group, sino yung nasa carboxyl group, ganito ginagawa dyan. So before the first amino acid, you write the NH3+. So that means ito yung N group, ito yung nasa leftmost side. On the end, you have the COO-. So that means you have the carboxyl group at the right side. Okay. Baka kasi mamaya baliktad yan eh. Okay. Pag baliktad yan, ibang amino acid yung tinutukoy natin pag ganun. Okay. So we have to be specific. Sino yung N group, sino yung carboxyl group. Okay. So let's try to write the single letter abbreviation for this molecule. Okay. So let's try to write the single letter abbreviation. Tyrosine, that will be Y. Saan kaya pwede? Burahin ko na lang tong part na to. Okay, so zoom ulit tayo. So gawin natin yung single letter abbreviation. So start ka, H3N+. Okay, so tyrosine will be Y. Okay, followed by threonine, T. Followed by glutamine, which is Q. Okay. Ang tawag ko dito sa glutamine pag single letter is cutamine, no? Kasi letter Q siya. Cutamine. Tryptophan is W. Isoleucine is I. And yan, COO minus na. Ito pa yung isa ko pang keyword, no? Para maalala ko kung bakit W yung tryptophan. Diba, this is the indole ring of the tryptophan. You can create W dito, no? Kaya siya W yung abbreviation. Ito yung W dyan, no? Okay. And letter W. Kaya tryptophan is W, no? Yan, para maalala mo agad na pag W, tryptophan. Pag glutamine, cutamine. Okay, so letter Q. Okay. And isoleucine, I, no? Or ile, no? Practice tayo, no? Kasi tapos na yung ating lesson, no? Practice tayo sa paggawa ng amino acid sequence, no? Paano tayo gagawa ng peptide bonds, no? Practicein natin yun. Okay. So, bukas lang tayo ng document. Daming calculations. Okay. So, practice tayo, ha? What if gawin natin ito? Bakit Chester nilalagay ko? Kasi prof ko dati si Sir Chester. Ito yung example niya madalas. Wala naman letter J dito, kaya hindi ko magawa yung pangalan ko. I pati M meron, pero J wala. No? Okay lang. Ayan, so try natin. Let's try to make the amino acid sequence Chester. So anong gagawin natin dito? So first, you have to draw all the amino acids muna, no? So i-draw mo yung cysteine, histidine, etc. etc. No? So, okay, so I want you to open the PowerPoint, no? Okay, so open nyo yung PowerPoint, then look at this slide, no? Yung sa Guide to 20 Common Amino Acids. Ito. Siguro naman nasa laptop kayo, no? So open nyo yan, then let's draw all of this, no? Ayan. Drawing natin. Letter C, we have the cysteine. So, paano ko ito drawing yan? So, nasa left side yung H3N+, followed by carbon, nasa taas yung COO-. Ay, hindi. Wag natin pala isulat sa taas yung COO-, yung H na lang. Then, dito sa right side yung COO-, kasi gagawa tayo peptide bond eh. C double bond O, OH. Siguro ang haba ng Chester, no? Palitan na lang natin. Igsian natin. Ang laki ng Chester eh. Ano kaya pwede? Letter C na lang umpisa, no? Kasi andyan na yun eh. Isip tayo. Ano kaya pwedeng word dito? 3 amino acids na lang muna para maigsi lang. Kunwari ito na lang. Palitan na lang natin from the top. Mahaba yung Chester. Gawin na lang natin is amino acid SAW, saw. Okay. So S, that means serine yan. Then A is alanine, then W is tryptophan. Okay, so yan. Madali lang muna, tatlong amino acids. Okay, so drawing natin yung serine. Serine is an amino acid that has ano, OH group there. Carbon, hydrogen, C double bond O, OH. Okay. So serine is ano, CH3OH, no? Ay, CH2OH pala. So this is serine. Then idudugtong natin yung alanine. So alanine is CH3 lang. So you write 3 and plus C H C double bond O OH, then CH3. Then lastly, you have your W or your tryptophan. Basa ko dyan is trypto1 kasi W1 na. Okay. Para mabilis ko maalala. C double bond O OH. Tryptophan, ito medyo mahirap i-drawing. Tignan nyo na lang kung paano yung illustration dito sa PowerPoint natin. So we have the CH2, C double bond CH. Okay, then you have your NH. Double bond here and your benzene ring. So yan, so we have the tryptophan na. Ayusin ko lang. Okay, so ayan. Ano susunod? Okay, so once you were able to draw your amino acids, ito yung susunod mong gagawin. Di ba yung carboxyl mo may H dyan? So ang assumption kasi dito is that all the amino acids exist in neutral pH, no? So that means yung ating carboxylic group ay deprotonated, no? So basically, ang gagawin mo lang dyan, tanggalin mo yung mga H sa carboxylic group. So yun lang, tatanggalin mo lang yung mga H sa carboxyl group mo. Okay? Tapos ano susunod dyan? Susunod mong gagawin, yung mga NH3, expand natin to show the bond. Okay? Pakita ko na lang. Pero kahit di nyo naman to gawin, okay lang. Gusto ko lang ipakita, no? Para makita natin yung peptide bond formation. So itong NH3, extend natin. Pakita natin yung mga bonds ng hydrogen to nitrogen. Then ano susunod? Ang susunod will be ganito. Yung dalawang hydrogen ng nitrogen at yung isang oxygen ng carboxyl group, that will be dehydrated, no? To form the water. And then yung carbon and yung nitrogen, dun sila magdudugtong. Okay? So paano yan? So ganito. Buburahin mo yung dalawang H. Ito, pati ito. Yung oxygen, burahin mo rin yan. Tapos pagdugtungin mo sila. That is now your peptide bond, no? Then you have the water here. Okay, so since natanggal yung mga hydrogen kay nitrogen, pwede mo na burahin yung positive charge. No, gawin natin uli. No? So dito sa right side. So on the right side, you have your two hydrogens. Burahin mo. Pag binura mo yung mga hydrogen na yun, mawala rin yung formal charge ni nitrogen. Yung oxygen, buburahin mo rin. Then pagdugtungin mo yung carbon and nitrogen. So, yan. So, that is your peptide bond. No? So, yan yung itsura ng ating peptide bond with the sequence SAW, no? SW. Okay, so pwede mo burahin yung water, tapos pagdikitin sila para mas maganda tignan. Pwede mo na sila pagdugtong-dugtongin. Imagine nyo yung katawan nyo, sandamukal na ganito yung meron doon. Okay. So this is our amino acid. This is our protein SAW. No? Ito yung serine, alanine, and the tryptophan. Okay. Ayun, ganun lang. Gawa pa tayo isa pang example. One example will be 4 letters naman. Isip ako. Ito pwede nyo to practicein ha. Practicein nyo na lang at home. Kunwari, gusto natin kunin yung ano kaya pwede? Read, no? Read. So, amino acid read, no? Yan ang pangalan ng ating protein. Nasaan yung mouse cursor ko? Okay. So, gawin natin yung amino acid read. So, R, EAD. So, asparagine. E is glutamic acid. A is alanine. D is aspartic acid. Okay. So, let us draw arginine, R. So, H3N plus. Yung OH, di ko nasusulat, O na lang kasi buburahin din natin mamaya. So, as arginine, CH2, 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 NH, C double bond N plus H2, and NH2. Okay? So, E is glutamic acid. So, glutamic acid, so that will be dalawang CH2 followed by carboxylic acid group. A is alanine. No, alanine, that is just an aliphatic amino acid. Aliphatic amino acid yan. So that means, ano yan, CH3 lang yan. Alanine. Then B is aspartate, no, or aspartic acid. So this is glutamate, however, with one carbon group shorter than the glutamic acid. Okay. Medyo nakakatamad to gawin, no? Isipin nyo na lang ganito yung ginagawa ng mga molecular biologists, no? Ito yung ginagawa nila to sequence the amino acid. Ayan, yung para ma-sequence yung amino acid ng protein. No? So, ayan. Imagine nyo yung COVID-19, no? Yung spike protein nun, around 1,000 plus na ganito, dugtong-dugtong yun. Okay. So next meeting, di-discuss natin kung ano yung effect ng paano nagkakaroon ng shape yung amino acids, no? So makukwento na natin yung COVID-19, yung sickle cell anemia, no? At iba pang mga disease, no? Usapang med tayo. So anyway, so this is our amino acids, READ, no? So arginine, glutamic acid, alanine, and aspartic acid. So again, to create the peptide bond, yung oxygen ng carboxyl, mawawala. Yung dalawang hydrogens nito, mawawala. Okay? So as a result, mawawala yung positive charge na formal charge kay nitrogen. So pwede natin sila pagdugtungin. That is one peptide bond. Okay? So ganun din mangyayari kay alanine. Burahin mo yung oxygen sa dulo. Bawasan mo yung tatlong hydrogen and nitrogen. Okay? Then pagdugtungin mo sila. Okay? So yan. Nakalimutan ko yung H dito. And then, ganun din yung gagawin natin sa last amino acid. Burahin mo yung oxygen dito. Burahin mo yung tatlong H. Palitan mo ng bond. Gawin mo ng peptide bond yon. Okay. So yan. So we have the peptide bond here and yung isang natirang hydrogen. Okay. So ito yung itsura ng protein na may sequence na READ. Okay. So kung gusto nyo gawin, kunwari marami kayong time, gusto nyo gawin yung COVID-19, yung sequence naman yan that is published naman, no? That is available online na. So it was published by China. Pwede natin gawin to, no? Pagdugtong-dugtongin natin. Pero okay na yun. Okay. So yan. Basta ang alam natin is that yung proteins, siya ay combined amino acids lang. And yung proteins natin nagagawa when we polymerize our amino acids, no? So ano yung tawag sa bond in between them? That's peptide bonds. Ito isang question, no? Suppose I want you to solve for the PI of the amino acid, no? Okay, kunwari gusto ko ipa-solve sa inyo ano yung PI ng amino acid, no? Okay. Kailan siya magiging Zwitter Ion? Actually, medyo mahirap 'yan kaya hindi ko ipapagawa sa quiz 'yan. Pero bigyan ko na lang kayo ng idea kung paano gagawin doon. Balik ulit tayo sa SO. Okay. So, suppose gusto natin kunin yung PI niyan ng SO. Balik ulit tayo sa PK table sa ating PowerPoint. Tinitignan ko na siya actually ngayon. Okay. So yung serine, anong groups yung nag-e-exist kay serine? Yung carboxyl group na gamit na to form the peptide bond. So ibig sabihin deactivated na yung alpha carboxyl group. Ang natitira na lang ay yung NH group, no? So serine, ang NH group niya ay may pK value na 9.15. Yung carboxyl group, hindi mo na siya magagamit kasi naka-peptide bond na yon. Okay? So, okay na tayo kay serine. Isa lang yung pwede natin magamit na value sa kanya. O, yung A, alanine. O, sa alanine, dalawa lang yung may pK value sa kanya. The carboxyl group and the amine group. However, for alanine here, highlight natin yung peptide bond para makita natin kung saan ako nakaturo. However, for alanine here, ito si alanine, yung carboxyl group niya, that is used for peptide bond. So, deactivated na yung COOH niya. Yung amine group niya, deactivated na rin. So, ibig sabihin, well, hindi mo na magagamit yon for protonization or deprotonization. Okay? So, wala na tayong magagamit na pK value for alanine. Okay? Kasi gamit na yung carboxyl and yung amine groups niya. So last one, we have the tryptophan. So for the tryptophan, kung titignan nyo yung table natin, dalawa lang din yung pK values niya. One is for the carboxyl group, the other is for the amine group, no? Tignan mo, ito yung tryptophan molecule here. Gamit na yung amine group. So we are now left with the carboxyl group of the tryptophan. So ito na lang yung pwede nating gamitin na pK value niya. So for tryptophan, the carboxyl group has a pK value of 2.2 0.38. So since dalawa lang yung pK values natin, our PI can be solved as the average of the two. 9.15, 2.38 divided by 2. So PI is now equal to 5.77. Okay. So at that pH, the protein SAW will precipitate. No? Doon siya mag-start mag-precipitate. Okay. So again, paano ginagawa? Tingin lang kayo sa pK values ng each amino acid dito. If the group is used to create the peptide bond, hindi mo siya isasama sa mga i-consider mo na pK values. For example, this carboxyl ng serine, ginamit siya for peptide bond, so hindi ko siya i-count. Okay, kay alanine, parehas ginamit for peptide bond, so hindi ko siya i-count. No? And so on, so forth. So try naman natin ito kay read, no? Kay protein na read. Okay, so isa-isahin natin yung mga pK values na pwede natin gamitin. So let's start with arginine. Okay, so arginine, tatlo yung pK values natin dyan. So kay COOH, gamit na. So hindi natin gagamitin na yun. So dalawa na lang matitira. Yung nitrogen group, which is 9.04. And yung kay R group, which is 12.48. Yan, so 12.48. E is glutamic acid, di ba? So glutamic acid, gamit na yung N group, gamit na yung C group, no? Carboxyl group for the peptide bond. So you are left with the R group na lang. So the R group is 4.25. Ito yung pK value niya, itong R group na to. Next one is the A, alanine. So again, katulad ng sinabi ko kanina, yung alanine, walang walang pK value yan. And then last one yung D. D is the ang tawag dyan, aspartic acid. So gamit na yung N group, ang natira na lang is the carboxyl group and the side chain group. No? So ano pwede natin gawin dito? So ito, kapag ganito na yung case, ang dami ng pwedeng ma-protonate, deprotonate. Ang ginagawa talaga dito is manual na chine-check yung yung Zwitter Ion, no. Gagawin mo doon, isa-isa mo sila i-deprotonate, no. Para mas makita mo kung kailan siya magiging Zwitter Ion, no. Let me do that na lang for you. Okay. So, kopyahin ko lang to uli lahat. Iisa-isahin natin to. Wala kasing shortcut kapag ganito nakalaki yung molecule. Ganito na lang. Hindi ko na siya i-rewrite, no? So, imaginein na lang natin. So, at very low pH, lahat ng carboxyl group, lahat ng amine group and yung R groups ay protonated if they can be protonated, no? So, that means itong carboxyl end protonated yan. Okay. So, yung amine end protonated yan and the R groups are all protonated. Okay. So, gawa tayo ng chart dito. 2.09. So, before 2.09 na pH, what is the overall charge of the molecule? So, walang charges dito except for the two nitrogen groups. So, that's positive 2. Then, as we go sa pH 2.09, what will happen? This tawag dito. This hydrogen hill will start to be deprotonated. So, magiging negative charge na yan. Okay? So, ang overall charge natin is from positive 2, magiging positive 1 na lang. Okay? So, what's next? Tingin tayo sa susunod na pH. Ito, 3.86 na. So 3.86 na pH, this carboxyl group will start deprotonating. Okay, so mag-start na yan ma-deprotonate. So hence, it will now become negatively charged. So may dalawa tayong negative, may dalawa tayong positive. So dito tayo magkakaroon ng Zwitter Ion. Zwitter Ion. Double T ata yun. Okay. Sunod tayo sa next pH. 3.86 followed by 4.25. No? So at pH 4.25, this carboxyl group will start deprotonating. Okay, so the overall charge of the molecule will now be negative 1. Okay. Tuloy natin. So, dagdagan mo ng dagdagan ng base until you reach the pH 9.04. At pH 9.04, this amine group here, tong amine group na to, it will start deprotonating. So, instead of being NH3, maging NH2 na lang yan. Mawawala yung positive charge for nitrogen, so the overall charge of the entire protein will be negative 2. And then lastly, we reach pH 12.48. So at pH 12.48, the nitrogen here sa guanidino group no, will lose proton so that mawawala yung formal charge ni nitrogen. So giving our entire molecule the overall charge negative 3. So for us to determine kailan yan magiging Zwitter ion, okay, so ito trinak natin yung changes sa protons, di ba, per different pH. So since ito yung Zwitter ion that is in between these two pK values, we can get the PI, no? So PI is equals to 3.86 plus 4.25 divided by 2. So kapag malaki na yung protein mo, you have to manually track, no, kung anong pK values magiging Zwitter ion. No? Unlike kapag isa lang yung amino acid mo, yung dalawang lowest, dalawang highest lang pwede. Pero kapag yan, sama-sama na yan, mahirapan ka. So, ima-manual mo talaga yan. So, this protein here, the protein read, will start precipitating at pH 4.06 na. Ganito yung ginagawa sa casein, no? Kaya nalaman na casein will precipitate at around pH 4 or something. Alam ko pH 4 yun. So, ginawa nila yun before is that sinequence nila yung amino acid and then eventually, dun sa amino acid sequence, inalam nila ano yung Zwitter ion, no? So, dun sa Zwitter ion, dun mag-start mag-clump yung proteins, no? Okay, so, ganun. Kasi if lumagpas ka sa Zwitter ion, magiging soluble uli yan sa water. Pag wala ka pa sa Zwitter ion, soluble uli yan sa water. No? So at this exact pH, dun siya mag-start mag-precipitate. No? Okay, so that will be all for our discussion sa amino acids. No? So ano yung mga na-cover natin today? So we covered that amino acids are the simple blocks, no, or the building blocks of proteins, no. And proteins, maraming function yan. Pwede yan yung maging enzymes ng ating body. Pwede yan yung ating mga cells such as red blood cell, protein lang yan. Okay? Pwede siya maging cellular membranes, no, yung proteins. Pwede rin naman siya maging muscles, no? Proteins din yan. No? So halos lahat sa katawan natin puro protein, no. And proteins are composed of small amino acids, no? There are 20 common types of amino acid, no? And may mga essential amino acids na kinukuha natin from the diet, no? Some can be created by the body, pero yung essential amino acid, di natin kaya gawin yon mag-isa, no? So ano yung mga essential amino acid? That is W, H, T, MILK, FBR, no? Ano yung mnemonic natin doon? William Howard Taft, Milk, and Fidel V. Ramos. No? So yun, WHT, FVR, MILK. So those are the amino acids that are essential. Okay. So there are 5 major groups no when categorizing our amino acid. Number 1 is the nonpolar aliphatic groups, pito 'yon. Sunod will be your polar amino acids, which is lima 'yon, 'di ba? Yeah, 5. So sunod doon will be your aromatic, which is 3. Okay? Followed by the positively charged R group, 3, and the negatively charged groups, 2. No? Okay. So, kailangan ba kabisaduhin yon? Kahit hindi na po kabisaduhin. Kasi you have the PowerPoints with you naman. Okay? Then, we can abbreviate our amino acid para mas mabilis siya maintindihan natin. So, instead of writing the entire name, we can just write the 3-letter symbol or the single-letter abbreviation. When our amino acid turn into protein, what happens is that they form the peptide bond. No? So the peptide bond is the linkage between 2 amino acids in a protein. The peptide bond is an amide bond and it has a partial double bond character because of the resonance in the amide structure. That makes the protein stronger. Okay? Kasi kapag di yan matibay, hindi tayo nag-exist ngayon. Okay? So matagal na tayong deads, no? Kapag ganun. And that also makes the peptide bond planar because of the sp2 hybridization of the atoms there, no? Due to resonance. And our amino acids are sequenced from N-terminal to C-terminal. So from N-terminal to C-terminal yung sequence niya. Okay? And yan, no. That will be all for today. Ang haba ng session natin ngayon. Na. So that's all for today. No? So expect that quiz will be available next week. Do you have any questions with our activity? So kayo na bahala, no? Pwede kayo mag-practice, no? Kunwari, gawan nyo ng structure yung pangalan nyo kung meron dito sa single letter abbreviation. Try nyo gawan ng amino acid yan. Okay? Then, kung gusto nyo hanapin kung anong pH mag-precipitate yung pangalan nyo, ganitohin nyo na lang yan. No? So, manually track kung kailan matatanggal yung mga protons ng inyong amino acids. No? So, yan. So, yan. That will be all for today. No? Yung ating assessment dito ay may multiple choice, no? Pero I'm thinking of, siguro magkaroon tayo ng parang case-based learning project for the, before the midterm exam, no? Malapit na naman kasi. Pwede doon yung mga proteins sa bahay, no? Let's try to see kung anong pH sila magpe-precipitate or not. Pero I will check, no, if that's feasible sa bahay. Titignan ko muna kung kaya ko gawin. Pag di ko kinaya gawin, edi mag-multiple choice na lang tayo. Maganda kasi yung mga experiment dito eh. However, nasa bahay tayo, so no choice. Wala tayong magagawa kundi mag-lecture lang. Pero I'll check if there are experiments that can be done at home. No? So kapag nakapag-collate ako ng medyo marami, hindi yan. Magagamit natin yan for the case-based learning project. Okay. Pero for now, focus lang muna kayo sa mga multiple choice questions. Okay. So yan. Do you have any questions, concerns, or okay na tayo? Medyo gutom na rin ako. Okay. So, nan na. So, with that, no? So, I'll see you again. Alam nyo, meron next week. Next week will be EDSA anniversary, Feb 25. So Feb 25 yon, ibig sabihin nun wala tayong pasok na naman. No? So okay lang naman. Okay. So okay lang yan. We still have 2 topics to go pero kaya naman i-discuss yung 2 topic in 1 week each. No? So we will still have plenty of time. No? Kasi ang midterms naman is 3rd week ng March. No? Ay hindi, sakto lang pala. So March na ulit tayo magkikita because of the holiday next week, no? So sa March 4, discuss natin yung 3D structure ng protein, and then sa March 11 yung enzymes na, no? Then after nun, we will have the midterm exam. Okay? So I'll see you again sa susunod na buwan, no? Sa March na lang. Okay? So aral maigi, and if you have any questions with this, no? You can ask me freely. Okay? So a copy of the class recording will be uploaded on YouTube, no? So bye-bye, and kita-kita na lang uli sa susunod. Okay? Ingat po.
Thank you, sir.